Yesterday we talked about the dietary fat composition, which comprises of mainly triacylglycerol, and the remain consist of cholesterol, cholesterol esters, phospholipids, and free fatty acids. So this slide shows the digestion of lipid in the stomach. No digestion takes place in the mouth. In the stomach, the dietary fat is acted upon by lingual lipase and gastric lipase, which target the triacylglycerol containing fatty acids of short or medium chain length, which is mainly found in the milk. So these have their own significance. We talked about this yesterday. Now let's move on to the next step of fat digestion, and that is emulsification of dietary lipids in the small intestine. This process, the emulsification, it takes place in the duodenum. The purpose of emulsification, which is basically breaking up of fat globules into smaller droplets. The big fat globule is broken down into smaller pieces. So the purpose of this is to increase the surface area of hydrophobic lipid droplets so that the digestive enzymes can act effectively. So the lipids are hydrophobic in nature and they are poorly soluble in the in the environment of the gastrointestinal tract. The digestive enzyme is also water soluble and can act only on the surface of the fat globules. So when you consume the fat in the diet, they tend to be aggregated together. They tend to come together and they make a big fat globule as it travels to the gastrointestinal tract. So to act the digestive enzyme, it has to act on this fat globule to bring about the process of digestion. But the digestive enzyme, the lipase, it can only act on the surface. So if it acts only on the surface and if the fat globules are when they tend to be together, then there is little space for the enzyme to act. But if the fat globule is broken down into smaller pieces, then the surface area increases. There is more area for the digestive enzyme to act upon, which makes the digestive process efficient. So this process of emulsification, it is brought about by two mechanisms. First is the detergent properties of the bile salts. Bile salts are very important. They are the most effective biological emulsifying agents. They are important for this process of emulsification. And some amount of emulsification is also brought about by the action of peristalsis, but bile salts are most effective. So in the exam, you might be asked to write about the role of bile salts in the digestion. So you are supposed to elaborate this process of emulsification. So the dietary fat, it is acted upon by bile salts, which are, I hope you remember the name of the bile salts: sodium taurocholate, sodium glycocholate, yatsai. So what they do, the bile salts, is they break down this big piece of fat into smaller droplets. Remember that when emulsification is being done, no bond is getting broken. Like you remember how triacylglycerol is made. There is glycerol and three fatty acids are esterified to it. None of those bonds are getting broken. Understood? No bonds is getting broken. Only big globule is being separated into smaller pieces. So this process of separation, breaking down of the big fat globule into smaller pieces, is termed as emulsification. Because of this emulsification, these tiny droplets are getting separated and are being acted upon by the fat digesting enzyme. So this is the fat, and this is the surface area where the lipase can act. Only this outer area. But when this is broken down into smaller numerous pieces, then the surface area increases. The enzyme can act on the surrounding of all these smaller droplets, which makes digestion efficient. So all along the surface of these tiny droplets, lipase action can start, and this is why emulsification is essential, and this process is being done by the bile salts. This emulsification, it makes sure that the surface area where the lipase can act is increased, and the bile salts also prevents the further coming together of these separated droplets. So in the beginning, the fat, it tends to be aggregated. Emulsification is carried out by the bile salt, and once these are broken into smaller pieces, bile salts also again make sure that these droplets again do not come together and become aggregated. So this is the importance of the bile salts in the process of digestion. You can make this figure as well when you explain the importance of role of bile salts in the process of digestion, which is emulsification. Now we have all the dietary lipids which need to be digested. We've got mainly triacylglycerol, we've got cholesterol esters and phospholipids which need to be converted to simple absorbable forms. Now, firstly, let's talk about degradation of triacylglycerol. It is degraded to 2-monoacylglycerol by the enzyme pancreatic lipase. From the molecule of triacylglycerol, two fatty acids are removed, which converts it to 2-monoacylglycerol, meaning there is only one fatty acid attached to the second carbon of the glycerol. So this is 2-monoacylglycerol, and you get two free fatty acids. Why is this necessary? Because triacylglycerol molecules are sorry. This is necessary because triacylglycerol molecules are too large to be taken up efficiently by the mucosal cells of the intestinal villi. The activity of pancreatic lipase is inhibited by bile acids, which are present along with the enzyme in the small intestine. So this is how triacylglycerol is degraded by the enzyme pancreatic lipase, which is present in the pancreatic secretion. Triacylglycerol is converted to 2-monoacylglycerol and two free fatty acids, which are the absorbable forms. There is another enzyme present, which is pancreatic colipase. This functions to prevent the inhibitory effects of bile salt on the lipase catalyzed hydrolysis of dietary long-chain triacylglycerol. So as I said, pancreatic lipase is inhibited by the bile salts. So this is prevented to make sure that lipase is getting like lipase can act efficiently. There is the enzyme pancreatic colipase present, which prevents the inhibitory effect of the bile salts on pancreatic lipase. So this is the second enzyme that necessary for digestion of the lipids. And another lipid that is present in our diet is the cholesterol ester. Do you remember what are cholesterol esters? Cholesterol plus one free fatty acid. When one free fatty acid is esterified to the position 3 of the cholesterol, you get cholesterol ester. Cholesterol, and this is the free fatty acid. So this cannot be absorbed. So the cholesterol ester, it needs to be converted back to cholesterol and release one free fatty acid. So cholesterol esters are hydrolyzed by pancreatic cholesterol esterase, which produces cholesterol plus one free fatty acid. So this is the enzyme that brings about the degradation of addition of cholesterol to cholesterol and one free fatty acid cholesterol esterase. So the fat digesting enzymes are first is pancreatic lipase, pancreaticolipase and cholesterol esterase. So as you have different lipids present in the diet, different enzymes are required. Like you need different enzyme for triacylglycerol degradation and different for cholesterol ester degradation. And you also have some amount of phospholipids present. Phospholipid is degraded by the enzyme phospholipase E2 and you get lysophospholipid and one free fatty acid which can be absorbed by the body. So with this, the fat digestion gets completed. The bile salts they play a very significant role in the digestion of fat because until and unless emulsification is done, fat digestion would get affected. So if there is some defect in the liver, if the bile salts are not getting formed properly, and if there is deficiency of bile salts due to any reason, in those patients, fat digestion becomes a problem because bile salts are needed for the fat to get emulsified. And if emulsification is not done, then proper digestion will not be carried out by the pancreatic enzymes. So this completes the digestion of fat. Now you have to know about the absorption of the final products as well. The final products of lipid digestion are mostly absorbed in the duodenum and jejunum. The luminal surface of the enterocytes is covered by a water layer which is unstirred water layer. So this water layer it represents the main barrier that the less soluble lipids must cross to be absorbed. These are the primary products of lipid digestion: monosaccharides, cholesterol, free fatty acids. So these need to cross the water layer before getting absorbed by the enterocytes. So these are the cells that absorb the primary products of lipid digestion, but they are being covered by this water layer. So these are not water soluble. So these need help to cross this water layer before getting to their site of absorption. So this is overcome by bile salts. Bile salts are necessary for absorption of the dietary lipids. So, bile salts are not only necessary for the process of digestion, process of absorption. But you remember that do not need the across this water layer. This formation is bile salts, long chain fatty acids that require the this micelles formation with the help of which the product with this figure bottom to micelle formation. These micelles are mixed micelles. These mixed micelles they are made of this cholesterol, these molecules are arranged in such a way that their hydrophilic and direct inward. This is the cholesterol. See how it is arranged. The hydrophobic cholesterol is present towards the inside, and the only hydrophilic portion of cholesterol, which is the OH group, is directed outwards. Same with monoacyl cholesterol, same with fatty acids, same with bile salts and phospholipids. So these mixed micelles are disc-shaped clusters of lipids that are coming together with their hydrophobic group on the inside, and their hydrophilic groups are present towards the outside. So this arrangement it makes the mixed micelles soluble in the aqueous environment of the intestinal lumen. Therefore, the bile salts allow the formation of water-soluble micelles, which transport the less soluble lipids to the apical surface of the enterocytes. With the help of this bile salt, all these products of lipid digestion, they are coming together and making this mixed micelle where the core is hydrophobic and the surface is hydrophilic. This arrangement helps all the products to cross this water layer and get absorbed by the enterocyte, which is the site of absorption. So this is the importance of bile salts in the absorption process as well. In the exam, you might be asked to mention the role of bile salt in the digestion and absorption of lipid. Make sure that you make this diagram. If it is difficult for you to make this one, you can just make this one and label it properly, and you write that bile salts help in the formation of mixed micelles. So okay, after formation of micelles, these approach the primary site of lipid absorption, which is the brush border of the enterocytes. So after the formation of micelles, all these products can then cross the unstirred water layer and get absorbed by the enterocytes. But short and medium chain length fatty acids, they do not require the assistance of mixed micelles for absorption. The long chain fatty acids, they are absorbed by the enterocytes, and the in essential fatty acids and fat soluble vitamins. The stool is difficult to flush because it floats due to the high fat content. So if you're asked to write a note on steatorrhea, you mention what is steatorrhea, you mention the quality of the stool, and you write that these are the causes of steatorrhea. The causes of lipid malabsorption are also the causes of steatorrhea. Okay, there is this disease in which there is fat is not digested properly, and it is cystic fibrosis. It is the most common autosomal recessive disease. It is a multi-system disorder which affects the pulmonary, gastrointestinal, and reproductive organs. In cystic fibrosis, there is a genetic defect. There is mutation in the gene coding for cystic fibrosis transmembrane conductance regulator protein. CFTR protein is defective. So you do not need to know the details of this. Just you know about this disease from the point of lipid dissection. So these are all the clinical features. Just go through it. There is salty, tasty skin. Salt is being excreted present in the sweat. High salt content in the sweat. The child has poor growth, poor weight gain, and severe respiratory disease and recurrent infection. And there is steatorrhea. So why is there steatorrhea and cystic fibrosis? Because due to the defect in the CFTR protein, the pancreatic secretions they become very thick. So when the pancreatic secretions become thick, they block the pancreatic duct. So when the pancreatic duct is blocked, all the pancreatic secretion they are not able to reach the duodenum. So you know that the pancreatic duct it opens in the duodenum. So when the pancreatic duct is blocked, pancreatic secretion they do not reach the duodenum. So when the pancreatic secretion when they do not reach the duodenum, there is absence of pancreatic enzymes. Okay. In cystic fibrosis, the pancreatic secretion they become very thick and block the pancreatic duct, due to which the pancreatic secretion does not reach the duodenum, which is their site of action. So when there is absence of pancreatic enzymes in the duodenum, digestion of fat does not take place because the fat digesting enzymes, the pancreatic lipase, is present in the pancreatic secretion. There is also defect in the digestion of proteins because all those protein digesting enzymes are also present in the pancreatic secretion. I hope you remember the names of the protein digesting enzymes: chymotrypsin, trypsin. These all are also present in the pancreatic secretion. So when the pancreatic duct is getting blocked, even the protein digesting enzymes are not able to reach the duodenum. So in cystic fibrosis, there is both defect in the digestion of dietary proteins and defect in the digestion of dietary fat. When the fat are not properly digested, they do not get absorbed and they are then passed in the stool, which is the cause for steatorrhea in cystic fibrosis. Diagnosis is sweat chloride test and genetic testing. When you do sweat chloride test, you can see that the sodium content of the salt is very high. So, okay, that is the absorption of the dietary lipids. So, the fat has gotten digested by the action of lingual lipase, gastric lipase, which does minimum work. And then, after emulsification, pancreatic lipase, cholesterol esterase, and phospholipase E2, they all act in the digestion of the different lipids. And then, with the help of bile salts, mixed micelles are formed, and the digested products are then absorbed in the enterocytes. In the enterocytes, there is again synthesis of lipids. Now, first, let's skip this fatty acid of short and medium chain length. They do not need the help of mixed micelles to get absorbed. They can just get absorbed by themselves. And even in the intestinal cells, they do not undergo any modification after absorption into the intestinal mucosal cells are then transported to the liver bound to albumin. So nothing happens to the short and medium chain length fatty acids. But the long chain fatty acids, there is some modification going on. I think this had been taught in your first year as well. Do you remember it? So this is the revision. So in the intestinal cells, what first happens is all those free fatty acids which were absorbed by the enterocytes, they are converted to their active form. Fatty acids are converted to their active form, fatty acyl CoA, by the enzyme fatty acyl CoA synthetase. You need 2 ATP to activate one fatty acid to fatty acyl CoA. So firstly, there is activation of long chain fatty acids. Look at this figure carefully. This is the enterocyte. This is the brush border where the absorption is taking place. This is the intestinal lumen where the fat digestion was taking place, and you get the products of the fat digestion. Like for example, let's talk about triacylglycerol. So this was digested and you got two free fatty acids and one monoacylglycerol. So these are absorbed in the enterocytes with the help of micelles. Okay. So in the enterocytes, again this triacylglycerol is remade. So you've got monoacylglycerol. Monoacylglycerol meaning there is glycerol plus one fatty acid esterified to it. So what happens then is another fatty acid comes and gets esterified to monoacylglycerol. Before that, fatty acid is activated, converted to fatty acyl CoA. This comes and gets esterified to monoacylglycerol. Enzyme is monoacylglycerol acyl transferase, and you get diacylglycerol. Diacylglycerol is glycerol plus 2 fatty acid. So one more activated fatty acid comes and gets esterified to diacylglycerol by the enzyme diacylglycerol acyl transferase, and you get triacylglycerol, which is glycerol plus 3 fatty acid. So this is how triacylglycerol is remade in the enterocyte. So you consume triacylglycerol in the diet, it is digested, converted to fatty acids and monoacylglycerol taken up by the enterocytes, and in the enterocytes itself triacylglycerol is remade. Same thing, there is resynthesis of cholesterol ester as well. Cholesterol, one activated fatty acid gets esterified to cholesterol. Enzyme is acyl CoA cholesterol acyl transferase, and you get again cholesterol ester. So similarly, phospholipids are also regenerated from the absorbed lysophospholipids. So this way there is resynthesis of the lipids in the enterocytes. In the enterocytes, the mixture of the absorbed lipids, they migrate to endoplasmic reticulum, and there is biosynthesis of the complex lipids. This is the summary of all those slides. Firstly, there is activation. The fatty acids are converted to fatty acyl CoA, and then two fatty acyl CoA, they get esterified to monoacyl glycerol, and you get triacyl glycerol, and the lysophospholipids are converted to phospholipids, and there is also esterification of one fatty acyl CoA to cholesterol, and you get cholesterol ester. So all these lipids have been regenerated in the enterocyte, but these lipids are hydrophobic in nature. They cannot travel in the blood by themselves. This triacylglycerol, cholesterol ester are completely hydrophobic in nature. They cannot travel in the blood by themselves. So do you remember how the dietary lipids are transported in the blood? Anyone? What is the transport vehicle for the dietary lipids?
Chylomicron.
Chylomicrons. Okay, good.
Chylomicron.
There is the formation of the chylomicrons in the enterocytes. Triacylglycerol and cholesterol ester, they remain in the inner portion, and they are surrounded by the phospholipids and apolipoprotein B48. So these chylomicrons, they are released in the blood. So these chylomicrons, they help to transport the dietary lipids into the blood from the enterocyte. The site of chylomicron formation is the intestine. So chylomicrons are then released, they move through the lymphatic system and the bloodstream to reach the tissues. So do you remember the metabolism of chylomicrons? Chylomicrons, when they reach the capillaries, they are acted upon by the enzyme lipoprotein lipase. What does this lipoprotein lipase do? It degrades the triacylglycerol that is present within the chylomicrons. The triacylglycerols are converted to fatty acids and glycerol. The fatty acids then enter the cell. In the cell, the fatty acids are oxidized as fuel, are used up or re-esterified and again made into triacylglycerol and stored. So this is the metabolism of chylomicrons. Not the complete metabolism that has already been taught in your first year. I hope you remember it. I hope you remember the metabolism of all the different types of lipoproteins. So this way the dietary fat has reached the tissues via the chylomicrons. So you should know about the all the enzymes that is required for fat digestion, the role of bile salts, and how they are again released in the blood. Please go through this case study. A 9-month-old child was brought with a history of poor growth and chronic cough. He has had multiple episodes of respiratory distress for which hospitalization was required. The mother has noticed that the skin of the baby tastes salty when she kisses him. The child also passes loose, greasy, foul-smelling stool. A sweat chloride test was done, which showed that the sodium content was more than 60 millimole per liter, which is more than normal. So, what is this child suffering from? Anyone?
Cystic fibrosis.
Cystic fibrosis. Can you explain the biochemical basis of greasy, foul smelling stool? Biochemical basis of steatorrhea. Pancreatic secretion are thick, which blocks the pancreatic duct. Pancreatic enzymes, which are required for digestion of lipids, are not reaching the duodenum. Fat digestion does not take place, and fat is excreted in the stool, leading to greasy, foul smelling stool. You should write this. This can be asked in your spotters in the practical exam. Okay, the you need to solve all these questions. This is for your homework assignment. List the steps and enzymes involved in protein digestion. Describe malabsorption syndrome. Describe the digestion of fat. Name the proteolytic enzymes and their role in digestion. Describe the gastric digestion of proteins. Please solve all these questions. In the malabsorption syndrome, uh, you write about uh, all the diseases that is found when the carbohydrate is not digested properly, protein is not digested, and lipid is not digested. So malabsorption, it shows that different nutrients are not getting absorbed. So you first describe where there is carbohydrate malabsorption, like in case of defect in the disaccharidases and lactose intolerance, there is maldigestion and malabsorption of the disaccharides. So in the protein, I give you a list of diseases where protein digestion and subsequent absorption was affected. So you write about those as well. And in case of lipid, you write the three conditions: pancreatic defect, defect in the bile salt, and defect in the small intestine. You list out all those, that will be sufficient. So okay, you complete all these assignments and you submit it to me when the classes resume. So you make a separate copy of biochemistry and you solve all these questions which I've given you. Understood? Okay, this completes our lipid digestion. In the next class, we'll talk about uh, biochemical changes, biochemistry of starvation. So, what I want all of you to do is you go through the feed fast cycle that had been taught in your first year. Understood? The biochemistry of starvation is mostly the repetition of the feed fast cycle. So, if you study the feed fast cycle and come for your class day after tomorrow, you will find it incredibly easy. So please go through it. Feed fast cycle. Understood? Okay. Goodbye, class.